Good morning. I'm Catera Wilder. I'm at UCSF, and I'm in the Bioengineering and Therapeutic Sciences Department. And some of the questions that I'm just broadly interested in is how do cells distinguish information? And specifically, during an immune response where there's many cytokines and chemokines that are in the environment, how do these cells know which ones are present and then also how to respond accordingly? And you can imagine having an accurate immune response is definitely important. And so that's really important. As you can see in the cartoon, I'm interested in the interferons. You can see there's a number of interferons that can bind to only 2 receptors. And so again, how do these cells distinguish which interferon is present? And as many of you probably already know, usually how information is transmitted in cells is there's receptors, and then they activate different signaling pathways, which then activate transcription factors that go to the nucleus to turn on different gene expression programs. And typically people just think that the presence of transcription factors are important for determining which signal is present. But I think that it's not just the presence of a transcription factor, but the dynamics of the transcription factor can help distinguish information. So again, I'm interested in these interferons. They typically activate the same pathway, but the dynamics of how they activate that pathway can be important for distinguishing which interferon is present. And so I've done a lot of work previously where we looked at some gene expression using RNA sequencing, where we could show that these interferons do have different gene expression programs that are activated. And they do activate this temporal code, if you can see down at the bottom. These interferons do have this distinct temporal code that cells are able to distinguish which cytokine is present. And we've also used a number of modeling techniques as well as experimental validation to try to understand the mechanisms that control some of these temporal codes. What I want to do next, or in our lab, what we're focusing on is trying to understand some of the extracellular factors, such as extracellular composition as well as extracellular density. How does that then change some of these temporal codes that the cells use to determine which interferons are present? And how does that ultimately impact immune cell recruitment? So again, these interferons are important for immune responses. How do those signals then tell how many immune cells should be attracted to a site of infection? And the overall goal of our lab is to be able to predict some of these immune responses, how many immune cells come to these sites for many diagnostic and therapeutic developments. And so if you guys are interested in that, I'm willing to talk, and thank you for your time.